Welcome to this Mtech Access webinar. At Mtech Access, we provide health economics and outcomes research and market access services from strategy through to implementation. Get in touch today to discuss your market access goals. First though, I hope you enjoy the webinar.
Welcome everyone, thank you for joining our webinar. It's called Surrogate Endpoints, Overcoming Reimbursement and Validation Challenges. We'll introduce ourselves in a moment, but let me run through the agenda. In today's webinar, we'll explain what surrogate endpoints are and how they're used in clinical trials. We'll also review the acceptability or not of surrogate endpoint data by HTA agencies, including any guidelines that have been given to sponsor companies. We'll look in depth at how surrogate endpoint validation is performed, and we'll illustrate this with a case study. Finally, we'll review learnings and look to the future and close with some information about how Mtech Access can support our clients in this field. So let's introduce ourselves first. My name's Claire Foy. I head the Global Market Access Strategy team at Mtech Access. Callum?
Hello everyone, thanks for joining. My name's Callum Jones. I'm a Senior Consultant Health Economist at Mtech Access of roughly 10 years, and I get involved in a lot of economic modelling, actually increasingly so within surrogacy validation exercises, such as which we're going to touch on today.
Hi there, my name is Laura Mathers. I'm a Senior Analyst in the Global Market Access team at Mtech Access.
Okay, so let's start by answering the question, what is a surrogate endpoint and why do we use them? A surrogate endpoint is an intermediate endpoint. It's intended to predict treatment outcome faster than a final patient-relevant outcome. One example is blood pressure, or another one would be LDL-C reduction in cardiovascular disease. Using surrogate endpoints can be beneficial in clinical trials, regulatory decision-making, and in real-world treatment. For example, early identification of treatment efficacy, so allowing for quicker assessment of the efficacy of a treatment before the primary clinical outcome is observed. Reduced costs and time, so we need fewer patients and less time to observe the surrogate endpoint than the final endpoint. Better patient outcomes, so surrogate endpoints may be more easily and frequently measured, allowing for more timely intervention and adjustment of treatment plans. And potentially, this enables healthcare professionals to make more informed decisions about treatment for their patients. Improved understanding of disease mechanisms. The study of surrogate endpoints can help improve our knowledge of the disease, which can lead to the development of new and hopefully more effective treatments. And improved chances of faster regulatory approval. Surrogate endpoints can provide evidence of treatment efficacy in a shorter period of time, which can help to accelerate the approval process and bring new treatments to patients more quickly. We will explore the use of surrogate endpoints in this webinar, including use in reimbursement decision-making. So using surrogate endpoint data has the potential to reduce the time required to gather evidence about an intervention. This means patients may have access sooner than they otherwise would. Surrogate endpoint data are accepted by many regulatory bodies, but HTA bodies and reimbursement agencies must also be prepared to analyse such data to inform their decision-making. To make this possible, it's necessary to validate surrogate endpoints and build confidence in their use. I'll hand over to Laura now to tell you more about the research we've done into the acceptability of surrogate endpoints by HTA agencies.
Thanks, Claire. So we conducted a targeted search of HTA agency websites to identify guidelines relating to the application or specific mention of surrogate endpoints. So following extractions, an assessment was then conducted to determine the level of guidance on the use of surrogate endpoints, which included the agency narrative on 4 main points. So it was the definition of a surrogate endpoint, examples of acceptable surrogate endpoints, methodological practices to support the use of surrogate endpoints in the relative efficacy or the health economic assessment, and the application of surrogate endpoints in a specific disease area or setting. We then summarise the overall level of detail in surrogate endpoints by HTA agencies, ranging from none, low to high, which you can see in the column to the right of the table. So from our findings, we found that the handling of surrogate endpoint evidence varied greatly across HTA agencies, with levels of evidence and statistical validation being inconsistent across markets. However, Australia, England, Germany and Portugal did lead the way in terms of published guidance on the use of surrogate endpoints. In England, we found that NICE didn't provide prescriptive advice on how to apply methods, only general commentary on establishing the surrogate to final outcome relationship. However, the DSU provided detailed recommendations regarding the use of multivariate analysis, including surrogate endpoint evaluation. And there have been positive decisions from NICE based on the use of surrogate endpoints, for example, in type 2 diabetes and PBC. In Germany, another high market, IQWiG and the GBA will consider surrogate endpoints if they have been validated with appropriate statistical methods, and they provide prescriptive advice on how to apply these methods. And then the markets with low overall detail of surrogate endpoint acceptance, surrogate endpoints have still been accepted. For example, the ZIN in the Netherlands have recommended drugs for IPF. The SMC in Scotland have recommended drugs in PBC. In France, another low market, the Transparency Committee, they don't provide prescriptive advice on how to apply methods, only general commentary on establishing the surrogate to final endpoint relationship. However, we found that surrogate endpoints will be accepted as a primary outcome measure on the condition that a link with clinical morbidity and mortality has been demonstrated. And then if we could move to the next slide, please. So following on from the previous slide, this map shows the level of published detail on acceptability of surrogate endpoints and how it varies, and there is a need for consensus across international HTA agencies. A current example of consensus across HTA agencies is UNETA in Europe, which published guidelines in 2015 on the handling of surrogate endpoints, representing a consolidated view of recommendations from experts across Europe. And these do appear to have been adopted by many European countries. However, they're not consistently applied. Okay, and now I'll hand over to Callum to tell you about how surrogate endpoint validation is performed.
Thanks so much, Laura. And the research that Laura was just referring to there is integrally important, really, to, of course, informing that health economists are adequately equipped, actually, to apply the most relevant and robust methods for indeed validating potential surrogate values for true endpoints. And to that end, what is actually currently the main basis of approach can be summarized in 5 key steps. Now, all these approaches, actually, any approach you find in essentially establishing a true surrogate, a valid surrogate, should begin with a comprehensive review of all relevant information, all data. So you begin actually really by identifying relevant clinical trials to your decision problem. And that decision problem is informed by the treatment ultimately that you're probably most interested in. That will be probably your new treatment, or indeed a treatment that is looking to strive further into the market post-launch. Now, we're going to be collecting relevant clinical trial data through typically double reviewer systematic reviews. We're going to be considering the collection within our PICO of relevant therapies, relevant patient characteristics, comorbidities, really trying to, of course, match a context that's befitting of our own within our data collection. We've been looking for and taking data from trials with at least 2 arms. Now, these are trial arms that contain data on both the endpoint values. So say we've got one surrogate of interest and one true endpoint of interest, for a simple example. Each trial that we include will have data on that surrogate of interest and that true endpoint of interest. It's also going to give us data on the treatment effect for both endpoints. So the treatment effect in that given trial on the surrogate and the treatment effect on the true endpoint. So we do require that, the 2 levels of correlation analysis we're about to touch on. We're also considering, as you might imagine in our PICO, again, trying to align ourselves in the best possible way to the context that we find our decision problem in. What are the publication date of the studies? Now, this is really pertinent frequently, because what we're thinking about is actually we don't want to be including studies that essentially predate a time and which predates actually where relevant treatment paradigms, the current treatment paradigm, is in play. So our decision problem at present concerns a certain treatment paradigm, a certain manner in which our treatments are perhaps administered and uptaken and responded to that aren't actually reflected in particularly old data. So publication data studies is an important substratification, sub-analyses to be running when we come to step 5 actually in these steps. So collecting that data early on, the appropriate data, is crucial. And we're also interested in sample size. So as you can appreciate, the greater the sample size of the data that we're collecting, the trials that we're collecting, the more accurate and easier it'll be to demonstrate validity, statistically significant validity of these surrogates for the true endpoints. And we might consider, of course, a certain minimum threshold of patients within a trial to be considered appropriate within our data pool. Step 2 really involves a meta-correlation between the reported values of the true endpoint and for the surrogacy candidate. Now, meta-correlation, all I'm saying there is we're taking all the data that we've collected within our SLR and we're running essentially Spearman-based weighted correlation analyses, essentially a weighted linear correlation of the data that we would plot. And we would plot data for individual arms of the collected studies, and these arms themselves would contain the data for the true endpoint and the surrogate candidate, these arms within each study. So we plot that data and we run Spearman weighted correlation linear analyses. Now, if that step proves or gives us a strong endpoint correlation, then we progress on to what we would call the treatment effect correlation. So this is the same sort of principle, but instead of taking data from our SLR extraction templates, which is arm level focused, which will give us endpoint specific values, we're actually interested instead in the trial effect data that's taken from those. So if we take the trial effect data from each of these studies and plot those, just as we did for the arm level data, so we can see the treatment effect on the surrogate and the treatment effect on the true endpoint. We plot those data points and again run a Spearman-based weighted linear correlation, meta-correlation, and we try and assess the strength of correlation there. If that's nice and strong, if that's looking good, in fact, if it's really good, we can almost stop there. We can almost stop there and say that what we have is a valid demonstration of surrogacy for this true endpoint. However, as I'm about to show, that's a high bar to exceed, and more commonly, if we have essentially a moderate strength of treatment effect correlation, so it's not too weak, but it's also not strong enough to preclude what I'm about to mention, we would estimate what's called a surrogate threshold effect. And we'll touch on that in a bit more detail in a second, but essentially that STE, surrogate threshold effect, is just a minimum amount of treatment effect we'd have to see on the surrogate to allow us to say, well, now we can this enables us to predict a significant benefit on the true endpoint. So it's a certain strength of effect we see on surrogate that would allow us to predict that significant effect on the true endpoint. Lastly, step 5, the scenario analyses. Well, correlations could be weaker, they could be stronger in certain subsets of data. As said, we might be actually collecting our data in our PICO on various bases. So thinking about our patient characteristics, our comorbidities, the therapies included, the data of these publications. So if we break down our analysis and see how the validity of our results holds up across these, and of course, as we get into more and more subsets, we're looking at smaller sample sizes. It's harder to find statistical significance of a demonstrated treatment effect correlation strength. But we may find interesting stories to tell, or indeed inferences to draw that would inform future activities and plans from those scenario analyses. So again, thinking about treatment class, comorbidities, patient characteristics, and so forth. So it's important to remember it's not just a base case estimation of surrogacy validity, but actually a subset as well. Thank you, Claire. If we could move to the next slide, please. Thank you. So here, hopefully to bring it to life a bit more for those watching and listening, we're thinking about an oncology context. Now, it's quite common actually in oncology, increasingly so, to be leveraging and exploring certainly the applicability, the validity of potential surrogates for overall survival. Overall survival in clinical studies in oncology, well, it's excellent news that patients are living longer and longer these days in oncology trials. There's nothing short of good news about that. It does, however, mean that if you can find any negative there, it's the fact that it takes quite a long time, of course, therefore, to understand the true treatment effect of a therapy. That actually to get overall survival data may take several years these days. which, as I say, is great news. On the other hand, it may mean we need to find a way, i.e. a surrogate, of collecting indication that the treatment effect is sufficiently strong sooner. So basically to be able to infer and estimate an improvement in OS at an earlier time than when OS is actually mature to collect. Quite often we're talking about progression-free survival. In this instance, we're looking at overall survival at 24 months. You can see here a plot which shows really all of our treatment arms. So all the clinical trials we collected in our SLR, sorry, I say arms, this is now treatment effects. So to be clear, these are actually the treatment effects that we can see, rather, of these trials. And we plotted all those treatment effects here. So we can see the treatment effect against OS, and we can see the treatment effect of the treatment in question on overall survival at 24 months. And what we're interested in is firstly the line. Now, of course, the slope of the line indicates the estimated treatment effect correlation. As it happens here, what we have is something that's rather strong. But what we're actually really interested in is the upper confidence interval of the estimated treatment effect. And in effect, we want that line and ultimately, therefore, the upper confidence interval to be as high as possible up that graph. Right? So we want the upper confidence interval to exceed actually 0.85, and we want the lower confidence interval to be as high as possible as well. So it just increases our confidence that we have a strong correlation effect. So we don't want to just take the base case correlation estimate, but actually the upper 95% confidence interval estimate. That's what we want to be considering. It has to be nice and high. And I would just say a couple of things of caution before we move on to the next consideration of surrogate threshold effect. And that is, as I'm sure many would remember from their biostatistics classes, if they took these, that we do have to consider the relationship may not be linear. So what we have here is a fairly fundamental example, illustrative example. It's relatively basic. If it's not linear, by extension, small improvements in overall survival at 24 months may not lead to commensurately small improvements in overall survival, or vice versa. We may have to be considering, based on clinician advice and indeed lessons from the literature, a different assumption there, something parametric, something that perhaps has a more exponential or logarithmic characteristic to the link between this surrogate and the true endpoint. And of course, as many are probably wondering and thinking out loud to some extent, this actually may be a relationship that isn't always the same for all patients. So some patients may respond really well to treatment insofar as they exhibit a benefit in the surrogate, say overall survival at 24 months, but actually not benefit in overall survival. Or vice versa. You might find that certain patients respond very well in an overall survival case, but actually their overall survival at 24 months isn't looking too rosy, and therefore actually we may have to consider that this isn't a holistic conclusion we could draw if we found a strong treatment effect correlation. If we move to the next slide, please, this is a consideration whereby, imagine if we, in this illustrative example, have found a treatment effect correlation that is of a particular strength, that isn't too weak to say, oh dear, we're going to have to stop here and abandon all hope of finding a true surrogate here between overall survival at 24 months and overall survival. But it's not so strong that we can say we can stop here because we've already proven it, essentially. And this is quite a common place to find yourself. And in effect, as I said earlier, the surrogate threshold effect is just telling us that a surrogate can accurately predict the true endpoint if the treatment effect on that surrogate is of a significant strength, of a sufficient strength, I should say. So a signal that's great enough from the treatment effect would then allow us to be able to say, we can actually use this surrogate to predict treatment benefit on the true endpoint of interest. And I think actually it would be great to just talk through this figure a little bit for your benefit and perhaps to bring the surrogate threshold effect to life. We can see here we've got two representations to form a linear assumption here between overall survival on the y-axis and overall survival at 24 months on the x-axis. Now on the left axis, the y-axis, overall survival, we have natural logarithmic of the hazard ratio of OS. Hazard ratio, probability of death, it's not a good thing. You want that to be as negative, as low as possible. On the x-axis, we have the logarithmic odds ratio of overall survival at 24 months, and that's a good thing. You want that to be as right-hand as possible. What we can do is draw a horizontal line that shows the point at which no benefit in the true endpoint of overall survival is shown. Right, so that would be where we find a value of 0 on the natural log of hazard ratio on overall survival. So on the y-axis, we can see the point at which that's 0. That's the point at which we're going to start seeing some benefit in the true endpoint of overall survival. And the STE, the surrogate threshold effect, we can draw from the x-axis. And that's the point at which our estimated upper confidence interval from the previously conducted metacorrelation analysis of treatment effects meets this threshold point. So we draw from the x-axis up to the tip of the upper confidence interval, and if that draws along onto 0 on the natural log of hazard ratio of odds of overall survival here, then that is the amount of effect we need to see on the surrogate to be able to then say this amount of, if we have at least this amount of treatment effect seen on the surrogate, this will allow us to use the surrogate to predict effect on overall survival, on our true endpoint of interest. So as said, negative values, as we go down the y-axis, of the natural log hazard ratio of overall survival are going to correspond to a treatment benefit of OS, and negative values of the natural log hazard ratio of overall survival are going to, as you can see, therefore, we get below 0, become negative, and they're going to meet our upper confidence interval where the odds of survival at 24 months exceeds the STE, right? So if we move on the x-axis rightward, then our line vertically up is going to hit the confidence interval, the upper confidence interval of our treatment effect correlation, and we follow that to the y-axis, and we're into the negative values of the natural log hazard ratio of overall survival. These, therefore, are treatment effects on the surrogate that would befit our ability to be able to predict benefit on OS. So therefore, we can see, this is, I hope, therefore clearer as to how one can see that the surrogate threshold effect shows the minimum amount of treatment benefit to be able to see, or that you have to see on the surrogate to be able to then infer, estimate a potential benefit in the true endpoint. I would like to say a few things regarding this. Economics is a social science, so is applied econometrics and biostatistics. There are caveats to be quite clear about, and any analysis would have to be upfront about these and as transparent as possible. This we've discussed so far is all about statistical likelihood, of course. This is not confirming causation, and indeed inherent to this, everything I've mentioned so far relates to the 95% confidence interval. It's a 95% chance likelihood, as we see it, a probability that the true value of the treatment effect correlation lies within these bounds. It's possible that it doesn't, right? So this is still something we have to take with a healthy, but not necessarily large, but a healthy dose of salt. Now, even if a treatment effect on the surrogate exceeds the STE, of course, therefore, this won't necessarily correspond to a benefit in the true endpoint. So we do have to be cautious in that respect. I think it's worth noting that within the field of medical statistics, this is actually still a hot topic of discussion and research. And it's just kind of an element, an aspect of watch this space, actually, because this is, albeit a fairly long-standing now set of principles used to validate surrogates, but the picture is always subtly changing. Some of the seminal work came out in 1999, followed by another iteration in 2007 by Boise et al. And that's just been iterated on thereafter and picked up by various HTA bodies, as Laura was mentioning earlier, and essentially refined on the basis of their recommendations. This is not a static topic. Anyway, at the moment, this represents essentially the gold standard in being able to predict the true endpoint value based on a validated surrogate. So there we go. And I wonder if we could just move to the next slide, please, Claire, and look at actually at a circumstance, a case study circumstance, where we had a client. This is not an atypical situation where they were looking to speed up the approval process for their new glaucoma treatment. And the idea is that it would be, in theory at least, quicker and simpler to collect data on surrogates for the true vision affecting endpoint in their trial than it would be to wait for the data to manifest for that true endpoint, right? So we can get data sooner on potential surrogates than we can for the true endpoint. The question thereafter, of course, as you'd imagine, was what are the surrogates that you could potentially consider as being plausible? And there's a few other considerations there as well. So we worked closely with the client's clinical ophthalmology team to narrow down essentially a breadth of plausible surrogates to shortlist the biomarkers that we deemed clinically plausible, that we agreed were being collected in the client's trial to a sufficient extent and quality. And they were likely to be routinely collected in clinical practice. And this is really important, of course, because going back to that first step that we mentioned, the systematic literature review, this is a key consideration if they're clinically normally collected, because this is going to be speaking to the likelihood of finding sufficient quantity and quality of clinical trial data that's going to allow us to be able to run through these steps that we've mentioned and to successfully validate biomarkers for the true endpoint of interest. So we, as said, followed the process that we covered 5 minutes ago, and ran that double reviewer SLR. We plotted all collected arm and trial level data into our respective two-step correlation analyses, ran these meta-correlation analyses, and estimated the surrogate threshold effect for what we found to be the single best performing surrogate biomarker based on its arm level correlation and its treatment effect correlation. What was really interesting was regarding this single biomarker, and actually it was found across a number of different potential biomarkers as well, that actually in our scenario analyses, you got stronger correlations for this biomarker between the biomarker and the true endpoint, the newer the data was. So the more recently the studies were published, the better it got, which is interesting because we were, by narrowing the field down to more new studies, the smaller the sample size ultimately that we were dealing with. So actually we found stronger, more statistical significance and better results the more recent the data. And this speaks to some extent to a change in treatment paradigm. It was felt, and there was a lot of ensuing discussion around how therefore to couch and caveat what we're dealing with here. Anyway, ultimately the study, the project, we feel proved useful in assisting our client to expedite appropriate evidence gathering for the purpose of gaining approval for their new treatments, and the process continues. But yes, hopefully, therefore, we have a good summary of how this is applied in practice. If we look at the next slide, what we see is some of the outputs there, and essentially you can see actually a representation of what I just mentioned. That the older the studies included, albeit more of them, we were actually finding weaker correlations at the arm and trial level than for later. And although I don't actually show it on this slide, you may just have to take my word for it, the statistical significance was greater as well for the more recent. And if we go to the next slide, please, Claire, we can also just see a temporal visual demonstration of that, as we go closer to 2023, our correlation coefficients are all the more inclined to be strong and significant. I think I'll just close there, Claire, but I'd be delighted to answer any questions that might arise relating to methodology around surrogacy validation. Thank you, Claire.
Thanks, Callum. Yes, please add your questions in chat if you have them. If we have time, we'll cover those at the end.
Okay, so now we've understood how surrogate endpoint validation is performed from Callum, I'd like to reflect on some key learnings from our research. So firstly, HTA agencies have stated a preference for final patient relevant endpoints. They also recognise that there is a need for surrogate endpoints to enable faster trials and access to treatment for patients. Secondly, HTA agencies expect sponsors to provide evidence to demonstrate the surrogate to final endpoint relationship, especially since surrogate endpoints do increase the level of uncertainty in coverage decisions. And then most HTA agencies did not provide detailed guidance on how to demonstrate the surrogate to final endpoint relationship or how to incorporate evidence from surrogate endpoints into the decision-making process. And then lastly, we found that few agencies have developed more detailed criteria in which they describe a meta-analytical approach to demonstrating the surrogate to final endpoint relationship. And if we could move to the next slide, please. So in terms of next steps, there is a need for HTA agencies to develop more detailed guidance for sponsors on the application of surrogate endpoints. And there may be an opportunity for clients to engage in this process to optimise success within HTA or reimbursement systems globally, which will ultimately see patients benefit from. And I'll now hand over to Claire to tell you all about how Mtech can support our clients in use of surrogate endpoint data.
Thank you, Laura. Hopefully you found that information interesting, and you may now be thinking about your own challenges and how you can use surrogate endpoint data in your trials and perhaps to achieve regulatory or reimbursement recommendations. The good news is Mtech Access can support you in your market access journey, including how to use surrogate endpoint data. For example, we can support evidence generation strategy and the gathering of evidence. We can help you consider whether to apply use of surrogate endpoints in your market access and pricing strategy. We can conduct landscape analysis to see how surrogate endpoints have been used in the therapy area of interest and any feedback from HTA bodies or other decision makers. We can support you in validating surrogate endpoint use via primary research with experts or advisory boards. We have a large network of contacts that we can use for that work. We can also, of course, support you in conducting the statistical methods that Callum's described. And finally, we can support with publications aimed at relevant healthcare professionals or payer audiences to ensure understanding and acceptability of the data. We have had a few questions, so thank you for sending those in, and we're going to take a few minutes now to answer a few questions before we close. Laura, perhaps you could answer this first one, which is, has this method been used in medical device or diagnostics assessment?
Yeah, so surrogate or proxy endpoints have been used in trials of interventions, which includes medical devices and a range of therapy areas. And there are examples of Mtech products being recommended by reimbursement agencies based on surrogate endpoint data.
That's great, it's good to know. Callum, maybe you could comment on this one. Could you possibly discuss the use of surrogate endpoints in the context of advanced therapy medical products?
Sorry, Claire, I was on mute there. You mentioned advanced therapy medical products, didn't you?
Yeah, can we use them in those high-tech products?
It's such an interesting question because now the need for surrogates in ATMPs is actually greater than you'd find out with ATMPs. This is because you have two major challenges frequently with this treatment type. Firstly, a real issue with small sample sizes. So, it's actually quite difficult to demonstrate statistically significant benefits on true endpoints for these type of products. This could be overall survival, as I mentioned previously. So, PFS and OS is a good example of this actually with oncology. But often these products are actually targeting diseases with longer than normal, to some extent, natural histories. This could take many years to show benefit in the true endpoint. So we're waiting a long time with not many patients to see essentially the kind of data we traditionally require to be able to demonstrate, for example, cost effectiveness or even just treatment benefit. So to that extent, I mean, the surrogate endpoints, biomarkers can play a really important role in the space of these therapies. There's a greater need, but at the same time, the challenge with these is to some extent twofold itself. The first is one, as all here would probably have thought already, that the small sample size problem doesn't go away with surrogate endpoints. To some extent, it's mitigated by the fact that there will be more patients at the beginning of the trial normally, so all else held equal, than towards the end of it. So some mitigation of that, but that issue still remains. And then secondly, actually in rare diseases, this is interesting. In rare diseases, particularly if there's no existing treatment, and this I'm glad this question's come up because it's so critical to the consideration of surrogacy validation. One cannot look to validate a surrogate or conclude the benefits of a drug based on a surrogate unless we're dealing with a surrogate that has been consensus agreed by relevant clinicians and indeed payers, HTA bodies, to be a plausible and appropriate surrogate, something that is conceptually considered to be of sufficient link to that true endpoint of interest. So with rare diseases without existing treatments, quite often we find there's almost no established consensus around the potential surrogates, and therefore there's usually a bigger step for advanced for ATMPs than otherwise in early on thinking, okay, are we actually even setting up the goalposts in a way that's fair and potentially fruitful? Now, clinical plausibility could be helped by certainly access to a good pool of KOLs and access to payers, as we touched on to some extent. This could be supported by extensive real-world evidence and registries. So real-world data out there that might, with observational study analyses that could be run, could relatively quickly start to draw up a statistical argument for, indeed, this is a plausible surrogate. So that's a consideration for demonstrating plausibility. But again, it goes back to, in these rare diseases, no treatments, clinical plausibility. Is it routinely collected? So are we going to have data on this in a non-observational real-world sense? And ultimately, of course, is it going to be collected in our trial? Now, quite often it's the case that we're thinking about surrogate analysis once the core trial, the key phase 3 trial has been set up and run. And ideally, you'd be thinking about surrogates potentially as a backup option earlier than this. So it is an important consideration as to whether we're even collecting data at our own trial that could be used to substantiate the surrogates that we're including, and also to thereafter, with our own treatment, client's treatment, predict the true endpoint of interest using that surrogate. So to summarize, we have a higher need really for surrogates within these advanced therapies, particularly rare treatments. But also it comes with its own challenges, introducing surrogates there. With respect to small sample sizes, something I would just add is that it's never an end game, so to speak, when you have a small sample size. It's a case when econometricians and biostatisticians roll up their sleeves and start rolling out relatively sophisticated missing data methodology or mitigating methodology. So small sample size issues can be worked around to some extent. It's not the end of the story, but it's all the more usually relevant a factor when we're talking about small trial advanced therapy investigations. So to summarize, I would say that the surrogates have a huge role to play in these, but at the same time, we do have these concerns. All the more reason and call for really rigor of methodological application, and of demonstrating actually the clinical consensus plausibility of those surrogates for the true endpoints of interest. These considerations just get all the more acute the rarer the disease is, and indeed the greater the paucity of existing treatments. So it's a rather big answer, Claire, but I think in effect, hopefully it touches a number of bases that might have also come up in different questions. That's why I wanted to cover quite a bit there.
Yeah, thank you. I think it did. I think that is a big question, and it's hard to answer it in this format, but of course, if anyone needs any further advice, please feel free to get in touch. It's certainly an area that we have expertise in. I think we've got time for one more question. So the final question is, what do you think it takes to move the needle on the acceptance of a surrogate endpoint in an HTA? So we've talked about this a little bit during today's presentation. I think the frustration is that some HTA bodies have not published any detail of what they consider to be an acceptable way of using surrogate endpoint data in submitting to an HTA body or another reimbursement agency. So we understand that using surrogate endpoint data does introduce uncertainty, and it can make it harder for HTA agencies to do their jobs. Obviously, they want to have confidence in the decisions they're making. But as we've seen from regulatory bodies, it is possible to validate these data and use them to accelerate drug development processes and approvals. And we would really like to see the same happening for reimbursement. The good news for patients is that there has been an increase in the development of methodological guidance for the use of surrogate endpoints over the last decade or so, and that's partly been driven by the adoption of the 2015 UNETA guidance that Laura mentioned. However, we feel there is room for improvement. So far, only a small number of HTA bodies have actually published any clear guidance on surrogate endpoint validation. We know that trial data is being submitted that have surrogate endpoint data as part of that, and we know that in some cases recommendations for reimbursement are being made on the basis of that, but it's quite inconsistent and not always clear how to get those data approved. So we would like to call for more development in this field. As Callum was saying, methodologically it's still developing, and we think we would like to see clearer guidance for sponsors. We'd also like to see more joined up thinking across HTA agencies and other payer organisations on how surrogate endpoint data can be used. Ultimately, we want to speed up access for patients to new drugs and we think on new interventions, and we think this is one way that that can be done. So on that note, I'd like to thank Callum and Laura, and to all of you for joining today's webinar. We'll send the slides from today's session later on this week. Please do contact us if you have any specific questions about our presentation or anything else that you think we could help with. The details are on this slide. So all that remains is for me to say goodbye for now, and thank you for your time, and we hope to speak to you soon. Thank you.
Thank you for watching. If you'd like to find out more about our work or how we could support your market access goals, please email info@mtechaccess.co.uk or visit our website at mtechaccess.co.uk.